In this video, we'll talk about another approach to clustering called hierarchical clustering. So a brief overview of this clustering method. In hierarchical clustering, we don't need to assume a certain number of clusters from the get-go, like we do in k-means clustering. And in hierarchical clustering, we use distances between cases to build what is called a dendrogram, which is a tree representation of the cases. This dendrogram represents a hierarchy of clusters in that clusters end up being nested within other clusters. And finally, nearly every implementation of hierarchical clustering uses what's called a bottom-up or agglomerative approach. That is, we start with every case as its own cluster, and we step-by-step join clusters together as we move up the hierarchy. This is actually the opposite of what we do for decision trees. So remember that for decision trees, we built the trees using the recursive binary splitting algorithm. And this is where we started with all of the cases in one group and we split the cases into different groups step by step. So let's look at a small example to get an idea for the clustering process before we look at the exact algorithm. So here we have a data set of 9 cases where we've plotted the values of the x1 and x2 parameters. So because hierarchical clustering is a bottom-up agglomerative approach, every case starts in its own cluster. We look at all possible pairs of clusters and find the closest pair in the first step, and this ends up being 7 and 5. So we fuse cases 7 and 5 together to create a branch. This is also called fusing 2 leaves to form a branch. In the next step, we again have to compute the distances between all pairs of clusters, but now we have to think about how to measure the distance between clusters when a cluster has more than one case. So there are actually different choices for doing this, and these different ways are called different types of linkage. We'll talk about linkage shortly, but suppose for now that we have a means of computing these distances between clusters. So it turns out that the clusters with the single cases of 1 and 6 end up being closest, so we'll fuse them together. In the next step, we fuse the 8 cluster to the 7-5 cluster so that the original 7-5 cluster ends up being nested within the larger 7-5-8 cluster. And we keep going until all cases are in one cluster. And finally, at the end of the process, we have a complete dendrogram. So another thing to note, the vertical height at which clusters are fused indicates their similarity. So the fact that the branch at the lowest height is the one connecting 7 and 5 indicates that they're the 2 closest cases. And the vertical height where case 8 fuses with the 7-5 cluster indicates the distance between case 8 and the 7-5 cluster as a whole, which may or may not actually be a distance between 8 and 7 individually or 8 and 5 individually. This will depend on the choice of linkage for measuring distances between clusters. So another important note is that horizontal distance does not indicate how close 2 cases are. So 9 and 2 look close in this dendrogram, but they're actually quite far apart when we look at the original data. So why this happens is actually an artifact of drawing these dendrograms. We could have swiveled the branch indicated here to show 2 on the right and the 8-5-7 cluster on the left. This wouldn't have changed a thing about the clustering, but it would have made 9 and 2 look farther apart, which they actually are. So with this background in mind, let's actually dive into the hierarchical clustering algorithm. So the first step in the clustering process is to compute a dissimilarity or distance measure between all pairs of cases. And very often it's Euclidean or diagonal distance that's used. But other measures can be reasonable and should be considered carefully before we actually start clustering. So at the start, all n cases are in their own clusters because this is a bottom-up agglomerative approach. And using our calculated distance measures between all pairs of cases, we'll find the two that are closest and fuse them to form a branch in the dendrogram. Now we have n-1 clusters. So then we continue to iterate this fusing process, and among these n-1 clusters, we find the two that are closest and fuse them. And we keep going until all cases are in one cluster. So an important thing to note is that this algorithm is greedy. Once clusters are fused, they stay fused. So in describing the hierarchical clustering algorithm, we've alluded to needing some way to measure the distance between clusters that have more than one case. So methods for clustered linkage give us choices for doing this. 4 common types of linkage are summarized in this table, so take a second to read over this. And we'll look at some pictures to get a better sense of these different choices. So in complete linkage, we measure the distance between 2 clusters using the maximum possible distance between a case in the first cluster and a case in the second. So in other words, we compute all pairwise distances and say that the distance between the 2 clusters is the maximum of these pairwise distances. In single linkage, we measure the distance between 2 clusters using the minimum possible distance between a case in the first cluster and a case in the second. So again, we compute all pairwise distances and say that the distance between the 2 clusters is the minimum of these pairwise distances. So a frequent pattern that arises with single linkage is that it tends to create very long trailing clusters. So what this means is that if we look at the situation on the right, we see that the 3 black-gray cases that aren't put into a cluster yet frequently get tacked on one by one. So they continually get tacked onto the orange cluster because the distance between the orange cluster and each of these subsequent cases is very small with this linkage strategy. So depending on the application and data context, this may or may not be sensible. So this is just behavior to keep in mind when we look at the results of single linkage clustering. In average linkage, we use the average of all pairwise distances between cases as a measure of the distance between clusters. In centroid linkage, we compute the cluster centroids, as we did in k-means, and we measure the distance between clusters as the distance between their centroids. So centroid linkage is often pretty popular because it's so interpretable. Centroids are very interpretable ways of measuring the center of a cluster, but it can actually result in undesirable behavior when we look at the resulting dendrogram. So let's take a look at an example. So the data here come from molecular biology research. Every case is a cell culture coming from various cancer cell lines, and the predictors are expression measurements for different genes. So we used hierarchical clustering with centroid linkage on this dataset, and this is the resulting dendrogram. So a very odd aspect of this dendrogram is this very prominent, what is called inversion. And this is where the left and the right clusters are merged at a height that's lower than either of the individual clusters. So this means that the centroids of the left and right clusters are closer together than the centroids of the subclusters that individually made up the left cluster and the subclusters that individually made up the right cluster. This isn't necessarily a bad thing, but it does make the dendrogram look weird, which generally people don't like. And these inversions don't occur with the other 3 types of linkage. This is the same dataset clustered with complete linkage, which uses maximal distance between clusters. And overall, the results look sensible in that the clusters are pretty well balanced in terms of the number of cases that get put in the left and right branches. Also, the clustering seems to recapture the natural groupings of types of cell lines. This is the dendrogram for average linkage. It similarly looks reasonable. We're seeing that, for example, on the right, all the colon cell lines are grouped together. And similarly for leukemia on the left, for example. Single linkage also kind of seems to recapture the natural groupings of the cell lines, but again, it has these long trailing clusters that we notice where one case at a time gets appended to a cluster at every step of the algorithm. So it turns out that for single and complete linkage, we can cut a dendrogram at a certain height to create clusters out of the branches that fall off. And this gives us nice interpretations about the distances between cases in the resulting clusters. Cutting trees resulting from centroid and average linkage actually doesn't have the same nice interpretation just because of the way that distances between clusters are calculated with those linkage methods. So to look at an example, let's look at the dendrogram built from complete linkage. We could cut this tree at a height of 130, and this would result in two clusters indicated by the rectangles. We can interpret this cut as saying all cases in a cluster are within 130 distance of each other. And this will be true for both of the broader clusters that result from this cutting. And for a dendrogram built from single linkage, we could cut the tree at a height of 90. This also results in two clusters. And we can interpret this cut as saying all cases in a cluster have at least one case within 90 distance of it. So let's end with some considerations for performing hierarchical clustering. The nice thing about this clustering approach is that we don't have to choose the number of clusters beforehand, which we do have to do in k-means. But it is up to us to choose the height at which we cut the tree if we're doing single or complete linkage. So considerations for how to cut the tree are very much guided by scientific context. And just like in k-means, we need to give thought to the distance metrics and variable scaling. Particular to hierarchical clustering is that we have to pick the type of linkage we want to use, which is also dependent on scientific context and what you want to get out of the clustering. So ultimately, clustering is a pretty variable procedure, and it's important to perform very thorough sensitivity analyses by trying out different methods and choices within those methods. And in the end, we want to try to obtain some sort of consensus result from all of these different types of clustering.